Okay, so now we've got all of our pieces in place. Changes in splicing enzymes and promoters and transcription factors and transposable elements and number of copies of genes, number of copies of whole stretches of genes. What wind up being the consequences of this? Back to the question brought up before. So what we saw the other day is sometime futz around with one single base pair of one single gene, and you've got somebody who's going to be dead at 3 months of age. PKU, phenylketonuria, one single gene could be a total, utter disaster. My God, instead now, thanks to these macro evolutionary changes, you change one single base pair, and as a result, you change 7 different genes. No, not 7, but you change more than one gene. You have one single base pair change, and if it's an exon that's used in a lot of different combinatorial ways, you've now produced mutated versions of a whole bunch of different proteins. One single change in a transcription factor, and you've invented an entirely new network of expression. What are the odds of stumbling into something there that is going to suddenly be great and wonderful in all those different areas of consequence? It's really unlikely. What have we just seen here? This is a very stabilizing mechanism for equilibrium. Equilibrium, long periods of stasis. What we've got is if a single base pair change is going to affect half a dozen different proteins or affect through a transcription factor entire networks, the odds are pretty lousy that you are just going to happen to stumble into one that works in all those domains, or at least works in enough of them that it doesn't do you in others. Most of these macro mutational changes are going to be bad news. Most of the time, in other words, there is stabilizing selection against macromutational changes. So what have we just gotten? We've just gotten a straight line. We've just gotten long periods of stasis. So when do the changes come? When you have some circumstance that is extreme enough that it doesn't matter that if this mutation, you've made 11D new types of proteins and 11D minus one of them are not great news, and the final one used to kill you. If the final one now is the trait that is going to save you, it is going to carry the weight of all the other proteins that are changed. If you get what is called an evolutionary bottleneck, if you get a circumstance of such severe selection for such a tiny subset of traits that basically the rule is it doesn't matter if you have that trait, it doesn't matter how much of a network you've changed, you're going to be one of the ones who come through and everybody else does not. And the evolutionary record is full of all sorts of circumstances where there have been selective bottlenecks where like 1% of a population comes out the other end of it because of some rare trait that they had, which carries all the macro consequences of that. For example, there was clearly some sort of bottleneck of selection that went on with cheetahs about, I don't know, a couple of thousand years ago or so. People have estimated more accurately because all cheetahs are so genetically similar to each other that you can transplant tissue, you can graft tissue from any cheetah on Earth onto another one and there will not be rejection. All of the cheetah on Earth are closely related descendants of what had to have been fairly recently, a couple of thousand years, a tidy remnant population that now is highly inbred, and you had some sort of selective bottleneck that went on at that time. Similarly, I can't remember when in hominid history, but the suggestion was at least a couple of points in hominid evolution, there have been points of selective bottlenecks where glaciers or comets or who knows what, where only a small subset of individuals with some traits that have been up until then neutral or unlikely to be useful because of these big macro consequences, suddenly only the folks who had it came out the other end. What are we beginning to describe here? Circumstances of rapid change. Circumstances of punctuated radical change there. Circumstances that begin to make conceivable punctuated equilibrium. So intrinsic in this huge complexity of macro evolutionary change in DNA, because of the structure, all of it, intrinsic in it are two critical things. One is the vast majority of the time make a change which changes some whole network, and it's going to be a disaster. The majority of the time there's going to be stasis, and also intrinsic in that is the ability to have massive macro changes. Any of these examples we've had here, where in periods of selective bottlenecks, you're going to get something like that happening. So, Gould and friends are absolutely right, all of that. Not necessarily, because again, this radical rapid change period, that's for paleontologists, for biologists, that's thousands of generations in some cases. Again, what counts as very rapid in one scientific discipline may not be the same for the biologist at the other end. Nonetheless, this winds up being a mechanism for long periods of stasis and very rapid change.